George, you're working, and George and Blake come up. George is doing his postdoc in Shoreline, and Blake is a rising sophomore at the University of Memphis. Blake, how do you like the summers in Seattle?
Way better than Memphis. All right. Good afternoon, everyone. My name is Blake Robinson,
My name is George Daily-Lyles.
and today we will be presenting on the utilization of AI drug screening and whole exome sequencing for new treatment options in recurrent high-grade meningiomas.
What is a meningioma? Meningioma is a tumor that arises from the arachnoid cap cells in the meninges, the membrane that surrounds the brain and the spinal cord. Currently, meningiomas are classified into three different subtypes based off of their histological and WHO Health Organization grades. Grade 1 meningiomas are benign, slow-growing tumors that are often resected without recurrence. Grade 2 meningiomas are atypical tumors with increased cellularity and have a higher rate of recurrence after resection. Grade 3 meningiomas are malignant, aggressive tumors with a high mitotic rate and are associated with poor outcomes in patients. All high-grade anaplastic meningiomas are grade 3. The current WHO grading scale includes NF2 gene mutations as all mutations for grade 1s, 2, and 3. And published research done in the Ferreira lab has identified hyperphosphorylation of the RB1 gene as a biomarker for proposed grade 1.5 meningiomas, where histopathology and genetic evaluation did not predict recurrence.
Our research focuses on a 27-year-old female patient who presented to the hospital with a grade 2 meningioma. She also received surgery for that meningioma. Surgical resection is one of two treatment options for individuals that have a meningioma. With an individual as young as her, 27 years old, and with the ability for meningiomas to recur so highly, it's likely that this patient will have to go under surgery multiple times in her lifetime. And so there's a need for a new treatment option. The second of the two treatment options is gamma knife radiosurgery, a form of radiotherapy. This is often used in combination with surgical resection, but less so as a sole treatment option, therefore limiting its usability in practice. So we set out to develop a pharmaceutical treatment option for individuals with a recurrent meningioma. You'll see this already with people with prolactinomas and dopamine agonists, albeit these are hormones. We are trying to go about it a little bit more precisely and look at it on the DNA level. Here's our general workflow that we'll be talking about today. We go from surgical resection, where we take the tumor from the OR, and then we create these cuboids out of the tumor, and then we treat them with 31 FDA-approved drugs. And then we give those to Fred Hutch to further our sample to 500 samples from 31. And then hopefully we can conclude a significant drug to then prescribe to the patient.
After resection, our approach takes into consideration the complex ecosystem that surrounds the tumor. This unique environment consists of signaling molecules like cytokines and growth factors, immune cells, blood vessels, and the polysaccharides of the extracellular matrix, as well as the tumor itself. All of these components make up the tumor microenvironment, which comes together to influence tumor growth, progression, and response to therapy. Focusing on this 3D tumor model rather than traditional 2D cell lines, we are better able to mimic the structural complexity in cell behavior within our experiments.
To create cuboids, we gather the tumor from the OR and we slice it horizontally using a vibratome and a razor. We then chop it vertically to create these 400 micrometer cubes, which you can see on the right under the microscope. And then we plate them into a 96 well plate, which you'll see on the left for drug screening.
On day 0 of our experiment, we prepare the cell viability assay by suspending the cuboids in HFH media to grow and maintain the cells. We pair that with a real-time glow bioluminescence media, and then we plate the mixture. On day 1, we assess the baseline cell viability and group the wells with similar viability, as seen here with these 2 purple arrows. And then we treat them with the 31 FDA-approved kinase inhibitor drugs outlined in the yellow on the second graph. Drugs like PP121, which target the platelet-derived growth factor receptor and regulate cell proliferation, cellular differentiation, and cell growth. From day 2 to 5, we continue to monitor the cell viability, and that data is collected and analyzed and sent off to our collaborators at Fred Hutch, where they feed this through their AI drug pipeline. And here we're able to visualize cell viability each day and its paired drug. We use this to correlate the luminescence to the drug effectiveness on day 5.
After the 5 days of drug screening, we have some results to analyze. As George mentioned, we have cell viability, which goes over how many cells in a given plate are alive. You'll see for each drug, there are 2 assigned ratios of similar cell viability at day 0. This is represented by the red and blue bars for each drug. For drug 2, you'll see a smaller cell viability, indicating a better effectiveness for treating the tumor. And then you'll see drugs like drugs 1 or drug 6 that have a lot of variability to them, suggesting that we need to do further samples for these, maybe n equals 3 or n equals 4. And then you'll see drugs like drugs 9, especially for ratio 2, that seem to almost promote tumor cell growth. This is something we would never want to prescribe to our patients. But yeah.
Big takeaways. Humans have over 500 protein kinases that influence tumor growth at various stages. Our personalized approach to recurrent high-grade meningiomas uses the 3D tumor model and artificial intelligence to narrow list of drugs, that 500 list of drugs down to a couple. Drugs like AZD3463 show promise, but challenges like tumor heterogeneity and the limited number of cuboids derived from the tumor itself give us a little inconsistency in breadth to our experiments. These limitations, though, they can be addressed by honing in on the pathways that our 2 selected drugs affect.
So watch this out. No, let me see. Hold on, hold on.
We are being joined on Zoom as well. So please try. But if we have any more outbursts, that's just support for us. So please join in next time.
Now that we know what drug to use, drug 2, for this specific patient, the 27-year-old female, it's important to understand that relying on tumor resection for genetic characterization is effective, yet it does not solve the problem we were trying to solve, which is circumventing surgery, or at least future surgeries. Multi-omics provides us that opportunity. Multi-omics is the biological analyses of various cellular functions like DNA, RNA, and protein. You have whole genome sequencing and whole exome sequencing for DNA, RNA sequencing for RNA, and chip sequencing for proteins. These allow us to hopefully identify biological processes that patients present with and that drugs can act on, so that we can go from 1 patient to 10 or to 100. And I would just like to take this opportunity to say thank you to Dr. Ellen Bogan and Mrs. Ellen Bogan, Jim Pridgeon, of course, and Julie Bold and Sylvia, and our PI, Dr. Ferreira, and co-PI, Dr. Parada, and everyone in the lab.
Yes, thank you all for being here and supporting us. Thank you for being here and supporting all of our other co-NSSP students. I'm happy to have joined you guys this summer, and I look forward to everything that we do together.